Hi all. Today I'll be handling Module 6 Advances in Food Technology. So in this particular chapter we will be discussing on Nutrigenomics, Nutraceuticals, Xenobiotics, Nanotechnology in foods, single cell protein and novel proteins. So before moving to the chapter, I would like to give a very brief on what you mean by the food technology. Normally, food technology, it is a science. This particular technology is mainly used based on the information we got from the food sciences for the selection, preservation, processing, packaging and distribution of the food. So normally this food technology affects the consumption of safe, nutritious and wholesome food. At the same time, this food technology explains why we should process our food, what is the need for processing that particular food, and also what are the various techniques used for processing the food. That is the technology which guarantee the product's quality. So in this particular technology, we will be discussing the future challenge and the potential solutions normally we faced during the process. So this food technology, the production to consumption food system is complex, and our food which is diverse, tasty, nutritious, safe and it is less costly. At the same time, it has become easily accessible. So the technological advancement should be accelerated in order to feed the growing population. So why this food technology is mainly for the processing of the food for our future use. So under this food technology, what are the various techniques which guarantee the product's quality? We should identify what are the effective techniques that guarantee the product's quality. Now we are moving to the next part. So the first one is Nutrigenomics. So what do you mean by the Nutrigenomics? It is a combination of a two term, nutrient plus genomics means gene. So this Nutrigenomics, it is also known as nutritional genomics. The Nutrigenomics is broadly defined as a relationship between nutrients, diet and gene expression. So Nutrigenomics, it is a relationship between nutrients, diet and gene expression. Normally this Nutrigenomics, it is the study of how diet may affect the expression of genetic information in an individual and how an individual's genetic makeup affect the metabolism and response to nutrients, other bioactive biocomponents in food. So actually, I can give you a few examples such as in the case of a person who is having the phenylketonuria. That is a condition where the amino acid phenylalanine is not metabolizing. So that is mainly due to the mutation occur in a single gene. So the gene is not giving instruction for the secretion of that particular enzyme which is responsible for metabolizing that particular nutrient. So studying how the diet may affect the expression of the genetic information in an individual and how an individual's genetic makeup affect the metabolism and response to nutrients and other bioactive components in food. So I hope you are clear with the example. An important aim of this nutrigenomic research is to study the genome-wide influence of nutrition with a specific focus on the role of metabolic stress in the genesis of the metabolic syndrome, the collection of phenotypes combining inflammation, metabolic stress, insulin resistance, and diabetes. So it is understood that with the increasing changes in food habits and lifestyles, people are becoming more prone to diet-related disorders. Therefore, there is an urgent need to boost more research in this field to help people in understanding the relationship between diet and health and to ensure that everyone benefit from the genomic revolution. So another one example I can give you is a lactose intolerance. Normally this lactose intolerance is mostly common in adults. That is the person who is taking the dairy products. It is not metabolizing in the condition, the person who is having such condition. So that is mainly due to the mutation of some genes which is responsible for the production of enzyme called the lactase. So if you identify which gene is having mutated, so that particular gene is not responsible to produce that particular enzyme for the metabolism of that concerned nutrient. So we can avoid, in the case of a phenylketonuria patient, we can avoid the diet containing the amino acid phenylalanine, and so we can save the person from the occurrence of the future damage or the problems. Next is Nutrigenomics, is establishing the effects of ingested nutrients and other food components on food gene expression and gene regulation. It will also determine the individual nutritional requirement based on the genetic makeup of the person as well as the association between diet and the chronic disease. So this Nutrigenomics will identify the genes involved in physiological responses to diet and the genes in which small changes called polymorphisms, and the influence of environmental factors on gene expression. So through the nutrigenomic, we can identify the particular genes which is mutated, and that particular gene is responsible for the various metabolic stress in that body. So through this nutrigenomic, we can identify the particular genes and identify the particular nutrient. So the better thing is we can avoid the diet or restricted the diet which causing that damage. Now we will see the 4 basic tenets of nutrigenomics, tenets or principle of nutrigenomics. The first principle is improper diet are risk factors for disease. Dietary chemicals that will alter the gene expression and or change genome structure. The degree to which diet influence the balance between healthy and disease states may depend on an individual genetic makeup. Some diet-regulated genes are likely to play a role in the onset, incidence, progression, or severity of the chronic diseases. Now next is Nutrigenomics or Genetics deal with a few though important concepts. The first point is specific dietary profiles can modulate the delicate balance between health and disease acting. The specific dietary profile that can modulate the delicate balance between health and disease acting either directly or indirectly on gene expression. So that is what I said earlier, like through the nutrigenomics we can identify the mutated gene which is responsible for the metabolism of that concerned nutrient. So we can avoid that diet, and we will stick on some specific dietary profile. So that will modify our condition. Now next is the individual genetic makeup, that is the presence of polymorphism in nutrient regulated genes will affect the individual risk of disease. Personalized diet which take into account individual genotype represent the ultimate goal of nutrigenomics or nutrigenetic studies, as they can lower risk in genetically predisposed individuals or population groups. Now we will see how does the diet affect our gene expression. Genes express themselves through proteins. So normally the genes will express its characteristics through the proteins. So enzymes are also basically proteins. So enzymes are responsible to get things started for the initiation of a particular metabolism. The enzymes are having some role. Our genome instruct ribosomes. Genome means our genes will instruct the ribosome. Ribosome is the cell organelle to produce many enzymes that destroy the toxins. Some foods such as cauliflower, broccoli, Brussels sprout contain chemicals that actually tell our gene to direct biosynthesis of these enzymes. In some individuals, genes give an unclear instruction. So in some cases, some food contains some chemicals. So they will communicate the gene for the direct biosynthesis of that particular enzyme. But in some cases, the gene will only give some unclear instruction for making an enzyme for the metabolism of that concerned amino acid or phenylalanine. As a result, what happened here is the level of amino acid will be shoot up, and that will causing brain damages. So if you identify the particular gene, we can give some restricted diet so that amino acid will stop the damage, if such things can be done if we detect the problem during the early infancy period. So next is nutraceuticals. Nutraceutical means it's a combination of a two term. One is nutrient, another is pharmaceutical. Nutrient means that is a we know it is a chemical substance that nourishing our body. It is a nourishing food component. That is what we call the nutrient. Pharmaceutical means it is a medical drug. Now, this nutraceutical name was coined in the year 1989 by the Stephen D. Fellis. He is the founder and chairman of the Foundation for Innovation in Medicine. It is an American organization which is located in New Jersey. Nutraceutical, it is a broad umbrella term that is used to describe any product derived from food sources with extra health benefits in addition to the basic nutritional value found in food. So nutraceutical, we can say that is the any product. Nutraceuticals are the substances. We can describe the nutraceutical as any product that derived from the food sources with the extra health benefits. So that particular product is having some extra health benefit in addition to the basic nutritional value found in food. A nutraceutical may be a naturally nutrient-rich food such as spirulina, garlic, soy, or a specific component of a food like omega-3 oil from salmon. So we can say these are the few examples for the nutraceutical. Now, what are the benefits of the nutraceuticals? So, nutraceuticals, it is help to improve the health, it delay the aging process, it prevent chronic diseases, increase life expectancy, or support the structure or function of the food. So next is Xenobiotics. Xenobiotic, it is a chemical substance found within an organism that is not naturally produced or expected to be present within the organism. It can also cover substances that are present in much higher concentration than are usual. Or we can say that xenobiotics are the chemical substances that are not nutrients for the body and enter the body through different routes like inhalation, ingestion, absorption, etc. So that is another definition of the xenobiotic. So it is a chemical substances which is found within the organization, but it is not naturally produced and or expected to be present within the organism. An example I can give you is drugs. Drugs such as antibiotics, they are considered as a xenobiotics in humans because the human body does not produce them itself, nor are they part of the normal food. Now the word xenobiotic point out a large range of artificial components such as chemicals from the chemical industries, drugs for medication, from the pharma industries, hospital, medical centers, and human and animal waste, pesticides and fungicides from the agriculture field. These xenobiotics are two types, exogenous and endogenous. Now, next is nanotechnology in foods. So, before saying something about nanotechnology aspects in food, we will see what do you mean by the nanotechnology stands for. Nanotechnology is also shortened to nanotech. Nanotechnology or nanotech. It is the use of matter or substances on an atomic, molecular and supermolecular scale. It is the use of matter on an atomic, molecular or supermolecular scale or a very ultra fine particle. Using of ultra fine particle for the industrial purpose. That is what we call the nanotechnology. Now what is the role of nanotechnology in food? So nanotechnology applications in the food industry can be utilized to detect bacteria in packaging or produce stronger flavors and color quality and safety by increasing the barrier properties. Barrier property means it includes the permeability of gases, water vapor, aroma compounds and light. All such factors are very important for maintaining the quality of a packaged food. Okay. So, nanotechnology holds great promise to provide benefits not just within food products but also around food products. Nano food describe food which is cultivated, produced, processed or packaged using nanotechnology techniques or tools, or to which manufactured nanomaterials have been added. So these are the applications of nano food in various fields like food or beverage, ingredients. Single cell proteins are dried cells of microorganisms which can be used as a dietary protein supplement. They are used as an animal feed and can be used for human feed as a protein supplement. Also called novel food and mini food. So what are the raw materials used for the single cell protein production? Normally it requires microorganisms that serve as the protein source and the substrate, that is biomass, on which they grow. There is a variety of both the sources that can be used for the production of the single cell protein. The biomass used can be plant biomass or organic biomass. The microorganism used belong to the group of algae, fungi and bacteria. A list of microorganisms used for the single cell protein production. So the fungi used for the single cell protein production is Aspergillus fumigatus, Aspergillus niger, Rhizopus cyclopia. Yeast includes Saccharomyces cerevisiae, Candida tropicalis, Candida utilis. Algae includes Spirulina, SPS, Chlorella, Prinoidosa, Contras crispus. Bacteria like Pseudomonas fluorescens, Lactobacillus and Bacillus megaterium. So the pictures of various microorganisms can also see. Now moving to the next part. What are novel foods? So we can define the novel food as a type of food that does not have a significant history of consumption, or it is produced by method that has not previously been used for food. Novel protein. Novel protein means protein derived from new sources. Novel protein are either isolated directly from plants or obtained by microbial methods such as from fungi or yeast. Few examples for the novel proteins include mushrooms, spirulina, yeast protein, byproduct of oil foods, food analogous, soy products, etc. So these are few examples of the novel protein sources. So that's all. So in this particular chapter we have discussed various terms and all. Okay, thank you.